Picture the open ocean, a realm so vast and empty that it swallows entire mountain ranges without a ripple on its surface. The blue stretches endlessly in every direction, broken only by the occasional white cap of a wave. Here, thousands of kilometers from any shore, the water column plunges deeper than Everest stands tall. There is no shelter here, no refuge, no safe harbor. Or is there? What if I told you that floating on this featureless void are cities, living, breathing metropolises built not of steel and concrete, but of flesh and chemistry? What if these cities were not just homes, but elaborate traps engineered over millions of years to perfection? The ocean's surface is not as empty as it seems, and the architects that build upon it are far stranger than anything humans have conceived. The Portuguese man-o-war drifts on the swells like a translucent balloon, its body catching the light in shades of electric blue and purple. At first glance, you might mistake it for a jellyfish, perhaps one that has inflated itself with air. But look closer, and the truth begins to reveal itself in unsettling ways. This is not a single creature at all. It is a colony, a floating city of specialized organisms called zooids, each one performing a specific function that the others cannot. Some digest food, some reproduce, some defend, and some hunt. They share a common gastric cavity, their bodies fused into a single functional whole. Born from the same fertilized egg, these zooids are genetically identical siblings that can never separate, each one utterly dependent on the others for survival. They are individuals that have sacrificed their independence to become organs in a larger body. A body that has no brain, no central command, yet operates with chilling efficiency. The most visible part of this living collective is the nematophore, a gas-filled bladder that can stretch over 30 centimeters long. This is no simple float bobbing aimlessly on the waves. It is a precision-engineered sail, its shape carefully sculpted by evolutionary forces to catch wind at specific angles. A specialized gas gland deep within produces a mixture of gases, carbon monoxide, nitrogen, and oxygen, that fills the bladder and keeps it buoyant. The crest that rises from the top acts as a sail, and here is where the engineering becomes truly remarkable. The Portuguese man-o-war can adjust the angle of the sail by contracting muscles in the codon, the structure that supports the nematophore. When threatened, it can even deflate the bladder slightly, lowering its profile in the water. This pneumatic skeleton, as scientists call it, functions much like a hydrostatic system, using pressure and controlled expansion to maintain its shape and maneuver across the open sea. But the true horror hangs beneath the surface, invisible to anyone looking down from above. From the ventral side of the colony, tentacles descend into the depths like fishing lines cast by something patient and ancient. These tentacles can extend more than 30 meters down into the water column, and some reports suggest they can reach even farther. They are not simple strands of tissue. Each tentacle is lined with thousands upon thousands of nematocysts, microscopic coiled structures that function as combination harpoons and hypodermic needles. When a fish brushes against one of these tentacles, the nematocysts fire in milliseconds, driving hollow threads into the victim's flesh and injecting a potent cocktail of venoms. The fish is paralyzed almost instantly, its muscles seizing, its ability to swim vanishing in a heartbeat. The tentacles contract, drawing the helpless prey upward toward the gastrozoids, the feeding zooids that lack tentacles of their own but excel at digestion. The division of labor among the zooids reveals an architectural philosophy that borders on alien. The tentacle-bearing zooids, called tentacular palpons, are hunters that never eat. The gastrozoids digest food they never catch. The gonadendra produce reproductive structures that will eventually detach and drift away to spawn, creating new colonies in distant waters. Some gonadendra even carry vestigial nectophores, remnants of swimming bells that no longer function but hint at an evolutionary past, when the colony's ancestors swam through the water column rather than floating on its surface. Everything is specialized, compartmentalized, optimized. There is no redundancy, no backup system. If the tentacles fail, the gastrozoids starve. If the nematophore deflates, the entire colony sinks and dies. This is a city built on the edge of catastrophe, held together by nothing more than biology and luck. And yet, this fragile architecture has conquered the world's oceans. Portuguese man-o-war colonies drift across tropical and subtropical seas, carried by currents and steered by their sails. They are asymmetrical by design. The zooids hang from either the left or right side of the nematophore, never both. Some colonies are left-handed, others right-handed, and scientists believe this variation prevents entire populations from being driven onto the same shores by the same winds. It is a population-level adaptation, a hedge against collective extinction. When conditions are right, these colonies aggregate into massive flotillas, sometimes numbering over 1,000 individuals. Imagine encountering such an armada. The surface of the ocean transformed into a minefield of translucent bladders and invisible tentacles, each one a trap waiting to spring. Fish populations can be decimated by these blooms, entire schools vanishing into the collective stomachs of the swarm. The life cycle of this creature adds another layer of strangeness to an already bizarre existence. Each colony is either male or female, producing either sperm or eggs but never both. During reproduction, the Gonadendra are believed to detach from the parent colony and drift away, releasing their gametes directly into the water. No one has witnessed fertilization in the wild. The early development happens somewhere in the depths, hidden from human eyes. A fertilized egg becomes a planula, a tiny larval form that begins budding new zooids as it grows. First comes the nematophore and a single protozoid, the primordial feeding structure. Then more zooids appear, gastrozoids and tentacular palpons arranging themselves in specific patterns determined by genetic instructions written millions of years ago. Eventually, the nematophore inflates enough to lift the developing colony to the surface, where it joins the drifting cities already scattered across the blue expanse. So the next time you stand at the ocean's edge and gaze out at that endless blue, remember this: the apparent emptiness is an illusion. Beneath the surface and floating upon it, invisible to casual observation, are cities built by creatures that have no right to exist. They are colonies masquerading as individuals, architects working with gas and venom and specialized flesh to create structures that drift on currents and hunt with tentacles longer than a school bus. They are fragile and deadly, simple and sophisticated, ancient and endlessly adaptable. The Portuguese man-o-war is not just surviving in the open ocean. It is thriving, building its floating cities on the void itself, turning the most hostile environment on Earth into home. And it is only the beginning. In the depths and on the surface, other architects are at work, engineering habitats from materials even stranger, building cities that would make the man-o-war seem almost conventional by comparison. The ocean's true engineers have been here all along, invisible until you know where to look. Imagine drifting through the twilight zone of the ocean, that dim realm where sunlight fades to a memory and the water takes on the quality of liquid space. Your lights sweep across the darkness, revealing nothing but suspended particles dancing in the beam. Then, without warning, you encounter something that should not exist. A structure the size of a small car, translucent as glass, hanging motionless in the water column like a palace made of fog. Its walls shimmer with an internal architecture so intricate that your mind struggles to process the geometry. Chambers within chambers, tubes and funnels and filtering membranes arranged in patterns that seem both organic and impossibly deliberate. This is not a jellyfish, not a siphonophore, not anything you have been trained to recognize. As your lights play across its surface, you realize with creeping unease that you are looking at a house. A mansion, in fact, and it is made entirely of mucus. The architect of this ghostly palace is the giant larvation, a creature belonging to the genus Bathocordeus. At first glance, it resembles a translucent tadpole, its body rarely exceeding 10 centimeters in length. It has a bulbous head or trunk, containing the basic organs of survival. A simple digestive system, a primitive notochord that lends it some structural rigidity, and cells specialized for a single obsessive purpose. Trailing behind the trunk is a flattened tail that beats in constant rhythm, never resting, never pausing. The creature itself is almost invisible, its tissues so transparent that light passes through with barely a distortion. But the house it builds dwarfs the builder by an order of magnitude. These mucus structures can exceed 1 meter in diameter, 10 times the length of the animal that constructs them. To put this in perspective, Imagine a human building a habitable structure 30 meters tall using nothing but secretions from their own body, completing it in under an hour, living in it for a single day, then abandoning it entirely to build another. The construction process borders on the miraculous or the nightmarish, depending on your perspective. Specialized cells on the larvacean's head begin secreting mucopolysaccharides, complex sugars bound to proteins that form a viscous, sticky gel. This mucus emerges in sheets and strands, and here is where physics and biology perform their intricate dance. The larvacean does not mold the structure with appendages or manipulate it with tools. Instead, it inflates the mucus like a balloon, using precise movements of its body and controlled pulses from its tail to shape the expanding material. The process takes approximately 1 hour from start to finish. Watch it happen in time-lapse, and you witness something that looks less like biology and more like sorcery. A creature conjuring architecture from its own body chemistry, expanding a single-cell layer membrane into a multi-chambered dwelling complete with functional filtration systems, intake valves, and structural supports. The finished house consists of 2 distinct filtration systems nested within each other like Russian dolls made of slime. The outer filter forms the mansion's walls, a coarse mesh designed to exclude particles too large for the larvacean to consume. Anything bigger than a few hundred micrometers bounces off or slides along the outer surface, denied entry to the interior. Inside this protective barrier hangs the inner filter, a marvel of biological engineering that would make a chemical engineer weep with envy. This inner structure features 2 fluted arms that funnel water and suspended particles toward a buccal tube, essentially a feeding straw that leads directly to the larvacean's mouth. The mesh size of this inner filter is astonishingly fine, capable of capturing particles as small as a few micrometers across. Bacteria, phytoplankton, bits of dead organic matter drifting through the water column like microscopic snow. All of it gets trapped, concentrated, and delivered to the waiting mouth at the center of this mucus labyrinth. The larvacean itself sits in a chamber within the inner filter, its tail beating approximately 60 times per minute in an endless, hypnotic rhythm. Each beat drives water through the structure, creating a current that pulls in the surrounding ocean and forces it through the filtration membranes. Scientists have calculated the flow rates using laser imaging and computational modeling, and the numbers are staggering. A single giant larvacean can filter between 1 and 5 liters of water per hour, straining it through mucus screens finer than most industrial filters. Over the course of a day, that amounts to filtering dozens of liters. Scale this up to population densities observed in productive waters, where dozens of these creatures might occupy a single cubic meter of ocean, and you begin to understand the impact. Within their preferred depth range of 100 to 300 meters, giant larvaceans can collectively filter the entire water column in as little as 13 days during peak abundance. They are biological water treatment plants, invisible cities of mucus working around the clock to harvest the ocean's drifting nutrients. But here is where the story takes its darkest turn, where the engineering brilliance collides with something that feels almost wasteful, almost cruel in its efficiency. The filters clog. Particles accumulate on the inner membranes, blocking the flow, reducing the filtration rate. For most organisms, this would trigger a cleaning behavior, some mechanism to clear the debris and restore function. The giant larvacean does not clean its house. It abandons it. When the filters become too clogged to function efficiently, usually after about a day, the creature simply swims out of its mucus mansion, leaving behind a structure that took an hour to build and represents a significant investment of energy and resources. Then it begins secreting a new house, starting the entire process over again. Smaller larvacean species repeat this cycle every few hours. The ocean is thus filled with abandoned mucus houses at various stages of decay and descent, ghostly structures drifting through the water column like discarded shells. These abandoned houses do not simply dissolve into the surrounding water. They sink. Laden with captured particles, they are denser than seawater, and gravity claims them quickly. Unlike smaller particles that drift down slowly over weeks or months, giving bacteria time to decompose the organic matter and recycle it in the water column, these mucus houses collapse and plunge toward the abyss in a matter of hours. They deliver intact packages of nutrients to the deep seafloor, bypassing the entire midwater ecosystem. This phenomenon has a name, marine snow, But that term hardly captures the reality of what is happening. Imagine it raining houses. Imagine a constant shower of abandoned architecture, each structure packed with food, descending through kilometers of darkness to feed creatures that never see sunlight. Sea cucumbers, brittle stars, deep-sea amphipods, they all benefit from this mucus manna falling from above. The giant larvaceans are not just filter feeders. They are ecosystem engineers, redistributing nutrients vertically through the water column at a scale that rivals the biological pump of sinking fecal pellets and dead organisms. So when you think of habitats, of architecture, of engineering, expand your definition beyond bricks and steel. In the ocean's midwater realm, where pressure and darkness make conventional shelter impossible, life has found another way. The giant larvacean builds its cities from the most unlikely material imaginable. Mucus secreted from specialized cells, inflated like a balloon, structured with precision, and deployed as both home and feeding apparatus. It lives in its creation for a single day, filtering thousands of liters through membranes finer than any human-made filter, harvesting the ocean's microscopic bounty with ruthless efficiency. Then it abandons the whole structure without a backward glance, letting it sink into the abyss to feed the hungry darkness below. This is not just survival. This is biological architecture elevated to an art form, a philosophy of disposability taken to its logical extreme. And somewhere in the depths right now, a translucent tadpole is beating its tail inside a mansion made of slime, pumping water through mucous membranes, living its brief, strange life in a palace it will abandon before tomorrow's light filters down from the surface above. We have seen architects that build with venom and mucus, creatures that construct floating cities from the chemistry of their own bodies. But what if the construction material was even more ephemeral? What if you could build a prison from nothing but your breath? In the cold waters off Alaska and British Columbia, humpback whales have mastered an art that blurs the line between hunting and engineering, between predation and architecture. They descend into the depths carrying nothing but the air in their lungs, and from that air, they forge weapons. These are not simple exhalations, not random bubbles rising toward the surface. These are calculated releases, precisely timed pulses of air that spiral upward in patterns so exact they could have been drawn by compass and ruler. The whales are building nets, curtains, cylindrical prisons with walls that have no substance yet cannot be penetrated. They are weaponizing physics itself, turning the basic properties of bubbles into a tool for mass capture. And the prey, schooling fish and swarming krill, have no defense against architecture made from breath. Watch it happen from above, from a drone hovering over the dark green water. The surface is calm, betraying nothing of the engineering taking place below. Then, without warning, a ring of white appears in the depths, rising slowly toward the light. It is perhaps 15 meters across, a perfect circle drawn in bubbles. As it ascends, another ring appears below it, slightly smaller, nested inside the first like the layers of a target. Then another, and another, each one tighter than the last, creating a spiral of ascending bubbles that form a three-dimensional cylinder. The structure takes about 80 seconds to complete from the first bubble pulse to the last. The whale, a solitary engineer in this case, has been descending while releasing these carefully metered breaths, starting at a depth of around 25 meters and working its way through multiple rings. Every ring is clockwise, every bubble pulse separated by precise distances that the whale controls through its swimming speed and the timing of its exhalations. This is not instinct alone. This is calculation, planning, a mind solving a complex three-dimensional problem in real time while swimming through darkness. The innermost ring is the masterpiece, the final trap within the trap. Its diameter is remarkably consistent across different whales and different feeding events, roughly twice the width of a humpback's gape when its mouth is fully extended. Scientists measuring these structures from drone footage and analyzing them with photogrammetry have documented this precision. The spacing between individual bubbles in this final ring is significantly tighter than in the outer rings, creating what researchers call the mesh size of the net. In the outermost rings, bubbles might be separated by a meter or more, creating a loose barrier that nonetheless defines the boundaries of the trap. But in that crucial innermost ring, the bubbles are packed close together, minimizing the gaps through which prey might escape. The whale has essentially created a funnel, wide at the bottom to capture as much prey as possible, narrow at the top to concentrate everything into a space small enough to engulf in a single lunge. It is engineering constrained by anatomy, a structure designed around the dimensions of the mouth that will consume everything trapped within it. But why does it work? Why do fish and krill, creatures that swim freely through water, treat bubbles as impenetrable barriers? The answer lies in behavior refined over millions of years of evolution. Bubbles rising through water create turbulence, distortions in the pressure field that most aquatic prey species instinctively avoid. For schooling fish like herring, a rising bubble curtain triggers a panic response. Turn away, stay together, move toward the center where your companions are. For euphausiid krill, the primary prey of humpbacks in Alaskan waters, the response is similar. Laboratory experiments have confirmed what whales discovered through trial and error across evolutionary time. Bubble curtains create effective barriers for these species, corralling them without physical contact. The prey are not actually trapped by the bubbles. They are trapped by their own behavioral programming, their own hardwired responses to disturbances in the water. The whale has essentially hacked their nervous systems, exploiting ancient reflexes to create a prison of fear from nothing but air. Now comes the strike. The whale has finished releasing bubbles and stopped its descent. It waits, suspended in the water column, as the bubble rings rise toward the surface. The prey, corralled by the outermost rings, mill frantically in the cylinder of bubbles, their density increasing with each passing second as they compress toward the center to avoid the rising walls. Studies using animal-borne tags with video cameras have captured this moment from the whale's perspective. A swirling mass of krill or fish, their concentration increasing sevenfold compared to natural patch densities, all of them trapped in a space the whale itself created. Then, approximately 10 seconds after releasing the final bubble pulse, the whale begins its ascent. It accelerates from below, swimming upward through the center of its own creation, through the rising cylinder of bubbles and compressed prey. Its mouth opens seconds before it reaches maximum lunge speed. The throat pleats, expanding to create a cavity that can hold more than its entire body weight in water and prey. The whale breaches the surface in an explosion of white water and bubbles, mouth filled, throat grotesquely distended. The trap has closed. Researchers have quantified the efficiency of this technique with startling precision. By comparing lunge events with bubble nets to lunges without them, by measuring the energetic cost of bubble production and the increased prey intake it provides, scientists have constructed models of the cost-benefit ratio. The results are remarkable. Bubble net feeding can increase per lunge prey intake by over 7 times compared to feeding on naturally dispersed krill patches. And crucially, this increase comes with no significant increase in energetic expenditure. The whale burns roughly the same calories producing bubbles and lunging as it would lunging without a net, but it captures 7 times more food. This is tool use in its most fundamental definition, employing an external mechanism to increase foraging efficiency beyond what the body alone can achieve. Except the tool is not external at all. It is manufactured from the whale's own exhalation, dissolved back into the ocean within minutes, leaving no trace except the satisfied predator and the depleted prey school. In some populations, particularly in Southeast Alaska and the waters around Antarctica, humpbacks have taken this individual artistry and transformed it into group warfare. Multiple whales coordinate their bubble production, creating larger and more complex nets. Some blow the bubbles while others vocalize, producing sounds that may drive prey toward the center or coordinate the timing of the group's ascent. Roles appear to be consistent within groups. Certain individuals always produce bubbles, others always vocalize, still others position themselves to herd prey or deliver the final strike. Watch footage of cooperative bubble net feeding and you witness something that looks less like hunting and more like military coordination, each participant executing a specific function in perfect synchronization with the others. The bubbles rise, the prey compress, and then multiple whales erupt through the surface simultaneously, their mouths forming a collective trap far larger than any individual could create. Some groups have been observed together for years, their roles unchanging, their techniques refined through repetition into something approaching cultural tradition. So here is what we must confront. Intelligence in the ocean has invented architecture not for shelter but for slaughter, not for protection but for predation. Humpback whales carry no tools, build no permanent structures, leave no archaeological record of their innovations. Yet they are engineers nonetheless, manipulating their environment with breath and behavior, creating temporary prisons from bubbles and exploiting the neurological wiring of their prey. They customize their nets based on conditions, adjusting the number of rings, the depth of deployment, the spacing between bubbles to match the prey type and density they encounter. They can exploit low-density krill patches that would otherwise provide insufficient energetic rewards, expanding their ecological niche beyond what their anatomy alone would permit. This is not just hunting. This is warfare elevated to an art, physics weaponized through millions of years of trial and refinement. And every time a humpback descends into the depths and begins releasing those precise spirals of bubbles, it is building a city of air, a floating trap with walls made of breath, a prison that exists for mere minutes before dissolving back into the ocean as if it never existed at all. The only evidence of its existence. The bulging throat of a whale breaking the surface, filled with the harvest of its invisible architecture. We have witnessed architects that build from mucus and breath, engineers that construct ephemeral prisons to capture prey. But what happens when the habitat itself becomes the predator? What if the sanctuary offered to desperate creatures is not shelter at all, but a slow-acting trap? In the open ocean, where any floating object becomes a magnet for life, some of the most dangerous structures are those that appear most benign. Consider Velella velella, a creature so delicate and beautiful that it seems incapable of harm. Its translucent disc floats on the surface, rarely exceeding 7 centimeters across, catching the wind with a diagonal sail that rises from its oval body like a tiny ship's rigging. Each one is blue and violet, catching the light in ways that make it shimmer like stained glass. Individually, they drift harmlessly across tropical and temperate seas, posing little threat to anything larger than plankton. But Velella do not always drift alone. When conditions align in ways scientists still struggle to predict, these creatures aggregate. They mass in flotillas that stretch across kilometers of open water, transforming the ocean's surface into something that resembles solid ground but conceals a darker truth. Approach one of these massive blooms from above and the scale becomes overwhelming. The water disappears beneath a carpet of living discs, each one oriented by its sail to catch prevailing winds, collectively numbering in the millions or possibly billions. The ocean has become a meadow of blue and purple, swaying gently with the swells. For fish and crabs living in the open ocean, where shelter is the rarest commodity imaginable, this floating mass represents salvation. They swim upward from the depths, seeking refuge under the discs, hiding in the shadows cast by countless overlapping bodies. Small juvenile fish press themselves against the underside of the raft, believing they have found protection from larger predators cruising the open water. Crabs climb onto the discs themselves, clinging to the nematophore, the gas-filled float that keeps each velella on the surface. Everything seeking shelter crowds into this false city, this illusion of safety floating on an indifferent sea. And here is where the horror begins to reveal itself, slowly, almost imperceptibly at first. Velella velella is not a neutral platform. It is a cnidarian, a cousin to jellyfish and corals and the Portuguese man-o-war we met at the beginning of this journey. Hanging from its underside are tentacles, short compared to its relatives but no less armed. Each tentacle bristles with nematocysts, those microscopic harpoons that inject venom on contact. The toxin is mild by cnidarian standards, insufficient to kill a fish outright or even cause noticeable paralysis in most cases. But consider what happens when a juvenile fish takes shelter beneath not one velella but hundreds. Each contact with a tentacle delivers a small dose of venom. Each brush against the hanging polyps triggers another injection. The fish is not fleeing because the pain is manageable, the paralysis incomplete. It continues to seek shelter, continues to press itself into the floating raft, accumulating toxin with every movement. Hours pass. The venom compounds. The fish's movements become sluggish, its reflexes dulled. Eventually it stops swimming entirely, drifting in the current, trapped in the very sanctuary it sought. The cnidarian polyps extend from the underside of each disc, and when the fish stops moving, when it drifts limp and helpless, these polyps envelop it. Digestion begins externally, enzymes secreted onto the fish's skin, breaking down tissues into nutrients that the Velella can absorb. The raft is feeding, not quickly, not dramatically, but continuously, processing the steady stream of creatures that mistake it for shelter. This is not active hunting. There is no chase, no strike, no moment of predatory triumph. This is passive consumption, a toll extracted from anything desperate enough to seek refuge on a floating meadow of stinging cells. And because the bloom covers such vast areas and persists for days or weeks depending on wind and current, the cumulative impact can be devastating. Entire cohorts of juvenile fish, squid hatchlings, small crustaceans, all drawn to the false promise of shelter, all slowly succumbing to the accumulated venom of 1,000 tiny stings. But Velella is gentle compared to what happens when the Portuguese man-o-war aggregates. We have already met this creature, already understood its individual architecture. A colonial organism with tentacles that stretch tens of meters down into the water column, armed with nematocysts capable of killing fish in seconds rather than hours. Now imagine not one man-o-war but hundreds. Imagine them drifting together, their bodies separated by mere meters, their tentacles overlapping in the depths below to create a three-dimensional web of venomous filaments. This is not a meadow. This is a minefield. The water between the colonies becomes a death zone. Every cubic meter potentially occupied by invisible tentacles waiting to fire their weapons at the slightest touch. Fish attempting to navigate through the bloom have no warning, no visual cue to reveal the danger. The tentacles are nearly transparent, visible only when light catches them at certain angles or when prey is already struggling in their grasp. Researchers studying these aggregations off the coasts of Australia and Florida have documented blooms of Portuguese man-o-war that number over 1,000 individuals, sometimes washing ashore in waves that paint entire beaches blue and purple. Before they beach, while still at sea, these flotillas create zones that fish actively avoid once they learn the danger. But the ocean is vast. Fish populations are constantly moving, and there are always new arrivals, always juveniles that have never encountered a man-o-war bloom before. The tentacles take their toll continuously. Schools of small fish blunder into the perimeter and scatter, leaving behind individuals caught in the filaments, their bodies already stiffening with venom. Larger fish, attracted by the struggling prey, sometimes follow and become entangled themselves. The bloom becomes a focal point of death, drawing in predators and prey alike, extracting biomass from the surrounding waters with ruthless efficiency. And the colonies themselves show no coordination, no awareness of each other. They are drifting separately, aggregated by coincidence of current and wind, yet collectively creating an architecture of danger that functions as if designed by malevolent intelligence. The triggers for these mass aggregations remain mysterious despite decades of study. Scientists have proposed various mechanisms: convergence zones where surface currents meet and compress, wind patterns that drive floating organisms into tight formations, reproductive synchrony that causes simultaneous spawning and subsequent larval development, creating cohorts that mature together. The truth likely involves combinations of these factors varying by species and location. What remains consistent is the outcome. Temporary floating cities that promise shelter but deliver death. Habitats that function as traps not through intention, but through the simple arithmetic of accumulated venom and overlapping tentacles. Creatures seeking any refuge in the open ocean's emptiness find these rafts and make a calculated gamble. Sometimes they survive long enough to grow and move on. Often they do not. The ocean's surface becomes a lottery where the prize is a few hours of shade and the cost might be life itself. So here is the lesson written in venom across kilometers of ocean. Architecture in nature is not always benign, and shelter is not always safe. The floating rafts of Velella and Phasalia are not built to attract prey in the way an anglerfish's lure or a whale's bubble net actively captures food. They are simply existing, drifting where wind and current take them, armed with the defensive and feeding tools their anatomy provides. Yet, in aggregation, they become something more dangerous than any individual. They become false sanctuaries, phantom cities offering shelter that slowly transforms into a trap. Fish and crabs and juvenile squid seeking any refuge from the open water's vast exposure find these floating platforms and press themselves into the shadows beneath, not recognizing that the ground itself is hungry, that every surface bristles with microscopic weapons, that the accumulation of small doses of venom will eventually overwhelm their systems. This is architecture as ambush predation, habitat as slow-motion slaughter. And somewhere in the Pacific right now, the wind is pushing thousands of translucent disks together, compressing them into another vast raft, another floating meadow that will shimmer in the sunlight and draw desperate creatures to their doom, all without intention, all without malice, all through the simple fact of existence in a place where any floating structure becomes a beacon to the desperate and the lost. We have explored architects that build from mucus and breath and venom, engineers that transform their own biology into floating structures. But what if the most elaborate habitats were built not by the living, but by the dead? What if the greatest cities drifting on the ocean surface were corpses transformed into islands? When a great whale dies in the open ocean, its story is far from over. The massive body, weighing tens of tons, does not immediately sink to the seafloor as intuition might suggest. Instead, something strange begins to happen inside the carcass. Bacteria colonizing the tissues start their work of decomposition, breaking down complex molecules into simpler compounds. This process releases gases methane, hydrogen sulfide, carbon dioxide that accumulate inside the body cavity, trapped beneath skin and blubber. The whale begins to bloat, its abdomen distending grotesquely as internal pressure builds. And then, defying the expectations of physics, the dead giant begins to rise, or at least refuses to sink. It floats, suspended at the surface or just below it, transformed from living creature into something else entirely. A temporary island of flesh and bone drifting on the currents, visible from the air as a dark shape just beneath the waves or breaking the surface like the hull of a sunken ship. The transformation from carcass to ecosystem happens with shocking speed. Seabirds are often the first to arrive, wheeling overhead before landing on the floating body. Gulls, petrels, albatrosses, they tear at exposed flesh, ripping away chunks of blubber and muscle. Their calls attract more birds, and within hours the carcass is surrounded by a feeding frenzy visible for kilometers. But the birds are only the beginning. Beneath the surface, sharks circle the body, drawn by chemical signals spreading through the water like an invisible beacon. White sharks, tiger sharks, blues and makos. They converge on the floating island and begin tearing at it from below, their powerful jaws designed to rip through even the toughest whale hide. The water around the carcass turns cloudy with blood and tissue fragments, creating a slick that spreads across the surface. And still the whale floats, its internal gases providing enough buoyancy to keep it from sinking despite the tons of flesh being stripped away. If the carcass remains buoyant for more than a few days, something remarkable begins to occur. Opportunistic organisms from the surrounding water start to colonize the floating island. Barnacle larvae, always searching for hard substrate in the open ocean, detect the chemical signature of whale skin and attach themselves to any exposed surface. Within days, clusters of barnacles begin to grow, transforming patches of whale hide into miniature reefs. Algae spores settling on the carcass find purchase and begin to photosynthesize, creating green films across the body's upper surfaces. Small fish, those same desperate creatures that seek shelter under Velella rafts and Portuguese man-o'-war colonies, find refuge in the shadow of the whale carcass. They hide beneath the floating bulk, darting out to feed on tissue fragments dislodged by sharks and birds. Crabs climb aboard, scuttling across the decaying landscape, feeding on whatever they can tear loose. The dead whale has become a mobile ecosystem, a floating island supporting dozens of species, drifting across hundreds or thousands of kilometers as currents and winds push it along trajectories determined by physics rather than biology. This is where the story becomes truly strange, where death transforms into something that transcends simple decay. Some whale carcasses remain buoyant for weeks, long enough for the barnacles to grow noticeably larger, long enough for algae to form substantial mats, long enough for fish to establish territories and feeding patterns around the floating island. The ecosystem becomes self-sustaining in miniature. Algae producing oxygen and organic matter, barnacles filtering plankton from passing water, fish feeding on both the carcass and the smaller organisms it supports. The whale has become architecture not through any living process but through the accident of decomposition, through the chemistry of bacterial gas production creating buoyancy that should not exist. And here is the horror and the wonder entwined. This island is temporary, its buoyancy a countdown clock ticking toward inevitable collapse. Eventually, the skin ruptures from internal pressure or shark damage, releasing the accumulated gases. Or bacterial activity slows as nutrients deplete, reducing gas production below the threshold needed to maintain buoyancy. When this happens, the island sinks. The descent is not gradual. Watch it happen and you witness something that looks almost deliberate, almost purposeful. The carcass tilts, water rushing into the ruptured body cavity, adding weight that overcomes buoyancy. Then it plunges, dropping away from the surface in minutes or hours rather than the days or weeks it spent floating. The birds scatter, calling in confusion. The sharks follow for a time, pursuing the sinking mass downward, but eventually even they turn away as the light fades and pressure builds. The fish that sheltered beneath it are left suddenly exposed, vulnerable, scattered across kilometers of open ocean with no landmark to guide them back together. And the whale continues its descent, falling through the water column, through the thermocline and into the twilight zone, then deeper still into total darkness. It settles eventually on the seafloor, sometimes at depths exceeding 3,000 meters, entering an environment so different from the surface that it might as well be another planet. But its role as habitat is not finished. It has merely changed location, changed function, changed the species it supports. The seafloor ecosystem that develops around a whale fall is one of the strangest phenomena in marine biology. The carcass lying on the abyssal plain becomes an oasis in a desert, a concentration of organic matter and energy in an environment where both are desperately scarce. Specialized species appear as if summoned. Zombie worms that lack mouths or digestive systems but dissolve bone with acid secretions and absorb the nutrients directly through their tissues. Bone-eating snails with symbiotic bacteria in their gills that derive energy from decomposing lipids. Sulfur-loving bacteria that carpet the bones in white mats and chemosynthetically produce organic compounds from the hydrogen sulfide released by anaerobic decomposition. This community can persist for decades, processing a single whale carcass through multiple ecological stages. First, the soft tissue is consumed by scavengers. Then enrichment opportunists exploit the organic-rich sediments around the skeleton. Finally, sulfophilic organisms colonize the bones themselves and persist until every last calorie has been extracted. A single whale fall can support over 400 species, many of them found nowhere else, creating biodiversity hotspots scattered across the deep seafloor like islands in an archipelago of death. But whales are not the only architects building floating islands from unexpected materials. Consider pumice, volcanic glass so filled with gas bubbles that it floats despite being stone. When underwater volcanoes erupt, they sometimes produce vast quantities of pumice that rise to the surface and form rafts covering hundreds or thousands of square kilometers. These stone rafts drift on currents for months or even years, and within days of formation, they begin to be colonized. Algae settle first, coating the porous rock surfaces with green films. Barnacles arrive next, cementing themselves to individual pumice chunks. Then come bryozoans, tiny colonial animals that build intricate branching structures, and corals, polyps that normally require hard substrate and find it here floating impossibly on the open ocean. The pumice raft becomes a mobile reef, a stone garden drifting thousands of kilometers from its volcanic birthplace, carrying its community of organisms across ocean basins. When these rafts eventually wash ashore or become waterlogged and sink, they deliver their passengers to new locations, potentially repopulating damaged reefs or establishing species in regions they could never reach through normal larval dispersal. So here is what we must understand. The ocean's floating habitats are not always built by living architects with evolutionary adaptations for construction. Sometimes the most elaborate mobile ecosystems arise from death and destruction, from decomposing whales and volcanic eruptions, from corpses that refuse to sink and stones that should not float. These temporary islands support communities as complex as any structure built from mucus or bubbles, yet they exist only because of accidents of chemistry and physics. A whale dies and bacteria produce gas, creating buoyancy where none should exist. A volcano erupts and produces stone filled with trapped gas, creating solid ground on liquid ocean. Life colonizes both without discrimination, transforming death and destruction into habitat, extracting opportunity from catastrophe. And perhaps this is the deepest truth of floating architecture in the open ocean. It is not the intention that matters but the outcome, not the design but the function. Whether built from mucus secretions or decomposition gases, from breath or from volcanic glass, these structures all serve the same purpose. They provide substrate in a realm where substrate does not exist, shelter in an environment without shelter, concentration of resources in a desert of dilution. They are islands in the void, temporary or permanent, biological or geological, living or dead, but all united in their defiance of the ocean's essential emptiness. We have journeyed through floating cities built from mucus and breath, from venom and decomposing flesh, from volcanic stone colonized by the living. We have witnessed architecture that traps and kills, shelters that betray those who seek refuge within them. But there remains one final category of habitat, perhaps the strangest of all, structures built from a material that has no mass, no substance, no physical presence whatsoever. What if you could construct a city from light itself? What if the habitat you created existed only as patterns of photons pulsing through darkness, visible one moment and vanished the next? In the deep ocean, where sunlight has not penetrated for millions of years, where darkness is so complete that your eyes remain forever dilated, yet see nothing, some creatures have learned to build with the only material available to them: bioluminescence. They engineer habitats from coordinated flashes and glows, from chemical reactions that produce cold light, from biological lanterns that draw prey and predators alike into patterns that exist only in photons and perception. Consider the pyrosome, a name derived from Greek words meaning fire body. These are not individual creatures but colonies, thousands of tiny tunicates called zooids fused together into a single hollow tube. Each pyrosome can reach astonishing lengths, with the largest documented specimens exceeding 18 meters from end to end, making them longer than most buses. The tube is open at one end and closed at the other, and the zooids arrange themselves with geometric precision across the outer surface, each one oriented so its intake siphon faces outward and its exhalant siphon faces into the hollow interior. Water is drawn in through thousands of individual zooids, filtered for plankton and organic particles, then expelled into the central cavity where it generates a jet propulsion that moves the entire colony slowly through the water column. But the truly remarkable feature is not the architecture of the colony itself. It is the light. Every zooid in a pyrosome colony is capable of bioluminescence, and under the right conditions, they synchronize their emissions into patterns that transform the entire structure into a living lighthouse drifting through the depths. Watch a pyrosome at night from a submersible and you witness something that seems to defy the laws of nature. The colony begins to glow, faintly at first, a dim blue-green luminescence spreading across its surface like sunrise over a curved horizon. Then the light intensifies, pulsing in waves that travel along the tube's length. Thousands of individual zooids flashing in coordination, so precise that the effect resembles electronic circuitry rather than biology. The patterns change. Sometimes the entire colony glows uniformly, a cylinder of cold fire hanging motionless in the black water. Other times waves of light ripple from one end to the other, creating the illusion of movement or direction. Occasionally individual sections brighten and dim in sequence, producing patterns so complex they suggest communication or signaling between different parts of the colony. Divers who have encountered large pyrosomes at night describe the experience in language usually reserved for religious visions. Cities of light materializing from nowhere, underwater auroras pulsing with rhythms that seem almost intelligent, structures so alien and beautiful that the human mind struggles to process them as living organisms rather than technology from some advanced civilization. The functions of this bioluminescence remain partially mysterious despite decades of study. Scientists have proposed several hypotheses, each supported by observation but none definitively proven. The light may serve as predator deterrence. When disturbed, a pyrosome flashes brilliantly, potentially startling or confusing predators long enough for the colony to escape. The synchronized patterns might facilitate communication between colonies, though what information they might exchange remains unknown. The glow could attract prey, drawing zooplankton and small crustaceans toward the colony where they are swept into the feeding currents created by thousands of synchronized siphons. Most intriguingly, the light may have no function at all beyond being a byproduct of the chemical reactions that zooids use for other purposes, an accident of biochemistry that happens to create beauty in the darkness. Regardless of purpose, the effect is undeniable. Pyrosomes create temporary cities of light in regions where light should not exist, architectural structures composed entirely of photons emitted by coordinated biochemical reactions, habitats that can be seen from hundreds of meters away yet have no more substance than a hologram. But not all bioluminescent architecture is benign. Not all structures built from light are simply beautiful phenomena to be witnessed and wondered at. Some creatures have weaponized bioluminescence, transforming it from passive display into active trap. Consider certain species of predatory fish and squid that have evolved elaborate light organs called photophores arranged in specific patterns across their bodies. These patterns can mimic the appearance of safe structures. A cluster of small fish schooling together, a patch of bioluminescent plankton indicating nutrient-rich water, even the glow of a pyrosome colony offering shelter. Small fish and invertebrates, seeking safety or food in the absolute darkness, swim toward these light patterns. They perceive structure where none exists, habitat where only predator waits. The lights intensify as prey approaches, reinforcing the illusion of safety, of congregation, of a place where other organisms have already gathered. Then, when the prey is within striking range, the predator's mouth opens and the lights extinguish simultaneously, leaving the victim in total darkness with no time to react before the jaws close. The anglerfish represents perhaps the most famous example of bioluminescent deception, though its tactic is slightly different. The modified dorsal spine that dangles in front of its mouth is called an elysium, and the glowing lure at its tip is the esca. This structure is not simply a light but a complex organ housing symbiotic bacteria that produce the bioluminescence through chemical reactions. The anglerfish can control the intensity and pattern of this glow, pulsing it in rhythms that mimic the movements of small prey or the bioluminescent flashes of potential mates. In some species, the esca is elaborately shaped, featuring filaments and protrusions that increase its resemblance to something worth investigating. Prey fish in the deep ocean, where food is desperately scarce and any light might indicate either something edible or a place where edible things gather, cannot afford to ignore these signals. They approach, drawn by instinct and hunger, and discover too late that the light is attached to a mouth lined with needle-sharp teeth angled backward to prevent escape. The habitat they perceived, the structure they thought they saw, was nothing but photons arranged in patterns evolution has determined are irresistible. The horror of this deception lies in its perfection, in the millions of years of refinement that have gone into these patterns. Every flash, every pulse, every spatial arrangement of photophores has been tested against the perceptual systems and behavioral responses of prey species. The patterns that work best, that most reliably draw prey within striking distance, are selected and passed to subsequent generations. The patterns that fail less often result in predators that starve and do not reproduce. Over evolutionary time, the light patterns have been optimized to exploit specific vulnerabilities in prey psychology and neurology. They are not random displays but carefully engineered illusions, false architectures constructed from knowledge encoded in predator genomes about exactly which light patterns will trigger approach behavior in which prey species. This is habitat construction at its most abstract, building not with matter but with information, with patterns that manipulate perception and trigger hardwired responses. The prey is not actually trapped by the light. It is trapped by its own inability to distinguish signal from deception, reality from illusion. So we return to where we began. The open ocean, that vast realm where shelter should not exist yet creatures have found ways to create it from the most improbable materials. We have seen cities built from colonial bodies and venom, from mucous membranes fine enough to filter bacteria, from bubbles exhaled with geometric precision. We have witnessed habitats constructed from decomposing corpses and volcanic stones, temporary islands that drift for weeks before sinking or dissolving. And finally, we have encountered structures built from light itself, from coordinated bioluminescence creating cities of photons in regions of eternal darkness, illusions of safety and congregation that sometimes reveal themselves as traps. The ocean has been engineering floating habitats for hundreds of millions of years, long before humans conceived of architecture or tool use. These biological engineers work without blueprints or conscious design, guided only by the selection pressures of survival and reproduction. Yet they have produced structures of astonishing complexity and sophistication. Some provide genuine shelter and facilitate entire ecosystems. Others are elaborate deceptions, false promises of safety that end in death. All of them remind us that architecture is not uniquely human, that the drive to create structure and habitat is fundamental to life itself, and that in the ocean's vast emptiness, any floating structure, whether built from flesh, gas, mucus, bone, stone, or light, becomes a focal point for biological activity, a temporary or permanent city in the void where life concentrates, persists, and continues its endless dance of predation and survival in the most unlikely of places.